This protocol allows users to study how the immune system produces antibodies to pathogens or commensal microbes. In addition, molecular pathways that regulate immunoglobulin gene diversification can be studied using genetically engineered mice. The protocol uses a combination of common molecular biology techniques, including PCR and Sanger sequencing, and does not require single-cell sorting or deep sequencing. The Peyer's patches can be easily confused with the surrounding fat tissue, so visual demonstration of this method may be helpful for those who are unfamiliar with this organ to identify its location and observe its dissection. Begin by laying the mouse on the dissection pad with the abdomen exposed. Generously spray the body of the mouse with 70% ethanol prior to making any incisions to sterilize the dissection area. Make an incision into the skin across the abdomen and pull simultaneously on both sides of the incision with fingers or forceps. Then cut the peritoneal cavity with scissors to expose the internal organs. Locate the small intestine and remove it by cutting below the stomach and above the cecum. Remove any connective tissue and fat linking the folds of the small intestine together. Examine the external surface of the small intestine for the Peyer's patches, which are small oval-shaped structures that appear white below a thin layer